Hi, my name is Jim Leffler. I'm a third-year resident at the Duke University Medical Center's Internal Medicine and Psychiatry program, and this is an Internal Medicine Chairs Conference that I'll be giving today. So I'll talk about mitochondrial disorders and specifically speak to mitochondrial neurogastrointestinal encephalopathy. At the end of the lecture, students should be able to recognize two mechanisms of inheritance that deal with mitochondrial disorders, specifically recognize two human disease states that come from mitochondria, explain why patients can present later in life than at birth, and explain specifically how mitochondrial neurogastrointestinal encephalopathy pathobiology functions to cause disease. So talking about the mitochondria, its function is oxidative phosphorylation, and it takes glucose and it makes ATP. And it makes 64 kilograms of ATP in a day in all the mitochondria in our body. And you can see that we have the outer mitochondrial membrane, inner membrane space, inner mitochondria, the matrix, the electron transport chain. You'll remember Krebs cycle, and then multiple different proteins throughout the process. And at all of these points, you could have some type of defect that will lead to disease and disorder. And you can see that this thing, the mitochondria, needs to function pretty smoothly in order to achieve its outcome. And even with its interactions with other organelles, such as the endoplasmic reticulum. So first of all, how common is the disease? So the epidemiology of the disease. So it occurs 13 per 100,000, and it's actually one of the most common, that makes it the most common inherited error of metabolism in the world. But that didn't mean much to me, so comparative epidemiology, just thinking about other disorders that we run across, I tried to pick things that are kind of genetically based. So we have 1 in 100 results in celiac disease, so pretty common. You can see that now with all the change in offerings for people with celiacs. 1 in 3,000 is cystic fibrosis. I know down here in our region, particularly at UNC, this is a pretty commonly seen disease, and then on our pulmonary service, but you can also think 1 in 3,000 is not that common. 1 in 1,500 phenylketonuria. We check that in all newborns and can treat it pretty effectively. And then a disease that we actually see fairly commonly on the internal medicine service is type 1 diabetes, but it's less common than the collection of mitochondrial disorders. So almost 24 per 100,000 compared to the 13 that we saw with mitochondrial disorders. So I want to go over just 4 key points that I felt like from mitochondrial genetics. There are other things that separate it from nuclear DNA, but these 4 things I thought helped separate, in my mind at least, mitochondria from nuclear DNA. So maternal inheritance. You can see that the maternal ovum has 100 times more than the sperm, and these are not drawn to scale, but it's just to kind of put that in your mind, that the sperm doesn't bring a lot of mitochondria, that they're already there present in the ovum prior to fusion and then the creation of the zygote. The next thing is heteroplasmy. So the ovum brings all the mitochondria, and within the ovum you might have some mitochondria that are bad, here designated as red, or good, here designated in green. And that those organelles split unequally, and they segregate to different cells by chance. And so here you end up with more bad, kind of this bad lineage being created down here versus this kind of good lineage created where you have mostly green mitochondria to red mitochondria. And there you have this concept of being able to create bad ones and good ones within the same, from the same cell and within the same organism. And we call that heteroplasmy. Now what happens, heteroplasmy has a cousin called the threshold effect, designated here by the red line. And the concept here is that, again, segregation, unequal mitotic segregation, leads to more bad than good going into this one, but it's only 50% here. Still not enough mitochondria to exert an effect globally on the tissue, but then this cell divides again unequally, and now you have 75% mitochondria. This goes on to develop its own lineage of cells. its own lineage of cells. And we can see that at some point you go from not expressing symptoms to expressing symptoms, and we call that transition the threshold event. And you can see that here with these cells of the skeletal muscle and neurons that are stained for the COX enzyme. If I remember correctly, the blue stain means that they do have COX, and the brown stain means that they don't. And you can see that there's different levels of blue, almost like how we demonstrate the blue states not voting as much for a liberal candidate or not on our political elections, but you can see these kind of gradations. Lots of blue here, less, and then none. And then once you have met the threshold, then the muscle itself starts to change, and you can then have expression. Same thing noted here in the neurons as we go from more expression to less expression. Last bit I wanted to say about, and again, these are not the only things that make mitochondrial DNA inheritance different than nuclear DNA, but that in quiescent cells, so G0 quiescent, meaning that they're no longer actively dividing, such as these two types of cells require a lot of ATP, and because of that, they'll then, since they can't split any longer to make more neurons, the neurons themselves will make more mitochondria, and the muscle cells also will grow larger and then require more mitochondria to fuel its energy demands. And then if those mitochondria are bad, they go on to make more bad mitochondria, and so the mitochondria keep building and replicating even in the quiescent cell. So these are four areas where the mitochondria can break down. I kind of pointed these out earlier. It has to do with the subunits of that electron transport chain, building the proteins within the mitochondria, here allowing in and out proteins from the inner matrix to the outer matrix, to the inner membrane space from the matrix, here into the mitochondria itself, and then mitochondrial dynamics. And I was going to show this one because I think this is kind of interesting in and of itself. So I was going to focus on dynamics and then mitochondrial expression and maintenance. So mitochondria itself, in terms of the dynamics, that's the first one, is mito or thread, and chondria or granule. And this comes from whenever the things were first looked at in the 1800s, I think it was late 1800s, they noticed under the microscope that it would go from a thread to a granule, and so it got this name mitochondria. And you can see that diagrammed here in this New England Journal, the drawing, that it goes from these small granules, it fuses with other small granules to make these long lines. And then the long threads can be taken, broken apart by endoplasmic reticulum, and they become, again, the small granules. And then this is important in terms of removing degraded mitochondria, breaking them up, and then going through the process of regeneration. This New England Journal video here, you can take that and look at it, and you'll see the mitochondria coming together. The red ones and green ones will make yellow ones, and it just kind of reiterates that fact that they're dynamic. And the reason that it's important that they're dynamic is that that's how this balance allows proper movement of the bad mitochondria away. So let's say this one becomes bad, and then it fuses with this one that's good. And then here, the mitochondria are intelligent enough to move all the bad stuff down to this end. So we maybe get to keep some of this outer shell, a lot of the inner matrix, and just get rid of, a little bit instead of the whole granule. And then it's taken in a plasmic reticulum, helps it, breaks off that bad portion now, and then that bad portion ends up getting eaten down here, or degraded, and then cycled back into the system. Whenever this kind of proliferation process gets out of whack, you can imagine it has implications in lung cancer, where they no longer fuse, so you just have the granules, and those granules build up and build up and build up. And so if we give back this mitofusin, you can cause them to fuse, and then you restore the balance. Another disease where the balance comes off is pulmonary arterial hypertension, which we see in our 7800, and again, it's this intimal proliferation occurs because there's just an imbalance of fusion and fission, and we can again give it and push down and cause it to regress. Similar dynamic mechanisms have been noted in Parkinsonianism, Charcot-Marie tooth, otic atrophy, and then cardiometabolic diseases. Specifically, I'll point out here diabetes and then ischemia and post-refusion injury. That the mitochondria fusion-fission mechanisms get messed up in the heart here and then in the liver and pancreas here, and that this leads to injury, scar, and malfunction. So mitochondria are involved in these diseases that we see really regularly on our services. And then so that was dynamics, and now I'll transition to a disease. It's not really of the electron transport chain. It was more a disease of the nuclear genetics is kind of what I wanted to talk more about. Sorry, I took a quick break there to update my slide. So it's actually about mitochondrial DNA maintenance and expression. But to do that, I just wanted to point out a couple of things. Interestingly, the mitochondrial DNA versus the nuclear DNA, you can see how the mitochondrial DNA here in red and then the nuclear DNA in blue, we have both nuclear DNA and mitochondrial DNA making up the electron transport chain. And so mitochondria can exist with both mitochondrial genetics and disease, and then nuclear genetics and disease. And these will follow Mendelian patterns, and these will follow those patterns that we discussed above, patterns previously discussed during this video. But just wanted to point out that a disease at any of these enzymes can cause disease. And then the things more generally that it causes are kind of systemic signs and symptoms. Things to think about are places with lots of ATP use, right? So in the eye, in the brain, and the one that's demonstrated there is actually kind of this caudate putamen region that's being affected. But you can also have strokes and then encephalopathy, which we saw in our patient. The heart here, as discussed, and it's because of cardiomyopathy and conduction defects from poor mitochondrial affects. The muscle, you end up with weakness, and this is actually a red, ragged fiber here, and that's pretty specific to mitochondrial disorder. So if you can get a muscle biopsy, then you can find it. And then intestinal pseudoobstruction. So you can see here the large stomach, and you end up with this gut dysmotility because the gut requires quite a bit of mitochondria to function and move forward, and you have both dysautonomia of the neurons firing and then also of the gut muscle themselves, and then peripheral neuropathy I also want to point to. Something else that's not listed here, but that I've read is quite common, is lactic acidosis. And you can imagine why that would be, in that we see an increased lactate in things like sepsis and ischemic bowel. And if you think about why that is, it's because we have a decrease in oxygen delivery to the tissues in these two diseases. In here, lactic acidosis is going to go up. Lactic acid is going to go up in the disorders of mitochondria, not necessarily because we fail to get oxygen to the tissue, but the mitochondria themselves fail to use that oxygen, and so then we have anaerobic respiration going on, which is bad and is just kind of a key to say that, you know what, there's something wrong with kind of the way we're producing oxygen here. Obviously, check to make sure you're not missing sepsis or some ischemic bowel, but it's definitely another indicator that there's something going on. So then on to the disease that I specifically wanted to comment on, which is mitochondrial neurogastrointestinal encephalopathy, which is a disorder of the DNA maintenance and expression. And so in the normal processes of a patient, we have thymidine and deoxyuridine. With this, thymidine phosphorylase takes the phosphate group, puts it on a deoxyribose group, and then we end up with thiamine and uracil. And these are used in DNA manufacturing by the mitochondrial replication enzymes. and proteins. And that, again, is in the normal. In mitochondrial neurogastrointestinal encephalopathy, you don't have that protein. So you build up those precursors, thymidine and deoxyuridine. In the normal functioning cell, these are just taken care of by the nuclear DNA processing system. So in a non-quiescent cell, so still going through the cell cycle as we saw above, still kind of processing, this gets processed out. We get more of this to allow the mitochondrial DNA. In a quiescent cell, so again, in the quiescent cell, and you remember that they continue to make new mitochondria in those cells, these build up. They don't have the benefit of the nuclear DNA thymidine phosphorylase mechanism being active, and so then our mitochondrial DNA end up with imperfect and disease from these toxic metabolites being present. And you build up numerous genetic defects within the mitochondrial DNA, leading to an inability to produce ATP. And it's going to affect those systems that we talked about that need it: GI, neurons, and heart, and that kind of thing, and muscle. And so our therapies try to target those toxins. I'm going to draw a line down here, and this is going to be the restore enzyme activity on this side and remove toxin on this side. So the way they're trying to restore enzyme activity is with gene therapy, stem cell transplant, and then I know this says Creon, that's for pancreas replacement, but similar medications are being considered for our mitochondrial disorder to give back our thymidine phosphorylase that has been lost. The other thing is an attempt to remove toxins, and you have hemodialysis and peritoneal dialysis. And the hemodialysis, while it helps for a brief period of time, it's been shown in studies that the toxic levels of thymidine and deoxyuridine come back quickly because you have so much mitochondria that are affected that it's hard to remove that toxin for a long enough period of time. Peritoneal dialysis a little bit better in terms of suppression of toxic metabolite, but as soon as the patient comes off of the PD, then their symptoms come back. And this has shown promise. One patient had good outcomes at 6 months. That's when the paper was published, and so I don't know how that patient did after that. These are my citations if you want to follow up on any additional information about mitochondria. I think that as we understand them more, they're going to play a bigger and bigger role in our understanding of more and more diseases as we start to think about ATP and oxygen utilization and on the cellular level as being a cause for disease. All right, thanks so much.